With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is, which of the following feature of vector is required to identify the transformed cells? Options given are selectable marker, ORI site, ROP, restriction site. So if we'll see any typical vector, a molecule, it should act as a vector if it is having the following components. First of all, any molecule, it should have which is supposed to act as a vector, should have ORI. ORI, it is being used for replication. It means the vector can replicate itself. It's the origin of replication. That is must for any vector to act as a vector. So first thing. Second thing, that vector should contain a restriction site. Restriction site should be present in any vector so that desired gene of insert that can be inserted into that. So without restriction site, that vector can't be cut, and then DNA or we can say that gene of interest that cannot be inserted. And third thing is the selectable marker. Selectable marker is required to detect the transformed, or we can say that recombinant molecules from the non-recombinant cells or non-recombinant molecules, because everything, if we'll see practically, if we'll think about the genetic engineering, as we are saying, it is being transformed, it is being not transformed, practically everything is present in a solution, and you just look at the solution, you will find simple buffer solution. There is nothing else which you can visualize through the naked eye. So to detect that whether recombination occur, means the plasmid has taken up the desired gene of interest, or it hasn't taken up the desired gene of interest, or we can say that whether it is recombinant or non-recombinant, that can be detected using the selectable marker. Every vector should have selectable marker. Here I am showing one example. It is just an example of a PBR vector. It contains origin of replication, it contains various restriction sites, and it contains two selectable markers, that is for the antibiotic resistance from ampicillin and tetracycline. So based on that, we can detect whether the cells has been transformed, because if tetracycline will be disrupted, so it will only grow in the ampicillin resistant, and similarly vice versa. So based on that we can detect, and there are different selectable markers are different. If we see, generally these are antibiotic resistance gene, that is one of the method to detect the recombinant from the non-recombinant. Second thing is the blue-white screening. Blue-white screening is also act as one of the important and widely used selectable marker with the help of enzyme beta galactosidase. So there are multiple other selectable markers which are used for detecting the recombinants from the non-recombinants, because otherwise we can't detect until unless a selectable marker is present. So statements say, which of the following feature of vector is required to identify transformed cell? Selectable marker, this is correct. ORI site we have already discussed, this is required for replication, not for the identification. ROP, this is completely altogether different, it is a repressor. And restriction site, that is required to insert the DNA. This is also incorrect. Correct option is our option A. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.